If you lift a rotting log in a New Zealand forest, among the pill bugs and beetle larvae, you might find something brown creeping around on long but sturdy legs. It will be small and be carrying in front of its body a pair of limbs that look grotesquely oversized, thick, spiny, and folded inward like the arms of a mantis. This is Sorensenella, one of the most obscure and least appreciated predators in New Zealand's ancient forests. It is a harvestman, but it is nothing like the spindly, daddy longlegs that most people picture. Harvestmen, or opiliones, are among the oldest terrestrial arthropods and have a basic body plan: a fused body, a single pair of eyes on a raised mound, and 8 legs. Within this ancient order, the armored harvestmen of the suborder Laniatories are the most species-rich and morphologically diverse groups, and it is here, among the short-legged, heavily built, tropical and temperate Laniatories, that Sorensenella belongs. Sorensenella is a genus in the family Trienonychidae. The family is distributed across fragments of South America, South Africa, Madagascar, Australia, and New Zealand, the remnants of the ancient supercontinent Gondwana, and its members are united by their powerful pedipalps, the pair of appendages flanking the mouth that in Sorensenella and its relatives have been transformed into formidable prey capture weapons. The genus Sorensenella was first described in 1903 based on the type species Sorensenella prehensor, meaning one who grasps, a reference to its giant weaponized pedipalps. The genus currently contains 3 recognized species: Sorensenella prehensor, bicornis, and rotara. All of them are only found in New Zealand. The body of a typical Sorensenella measures 3.5 millimeters, so very tiny. The pedipalps on Sorensenella, however, are approximately 6 millimeters long, nearly twice the length of the entire body. They are studded with spines along every segment. The trochanter bears a short upper spine and a long lower one. The femur is armed with 4 or 5 spines dorsally and 6 long spines on the underside, and the tibia carries its own spike array. In males, the pedipalps are even larger and more heavily armed than in females, a sexual dimorphism that hints at their importance in both predation and possibly combat for courtship. On its dorsal surface, Sorensenella species bear 1 or 2 conical projections, called spinous tubercles, on the sides of the body. The eye mound has a small apical spine as well. This ornamentation is not decorative. It likely serves as both a defensive function, making the animals harder to swallow for small predators, and as a useful taxonomic one, allowing specialists to distinguish species and subspecies. The life of a Sorensenella is spent almost entirely between soil and rotting wood. These are cryptozoic animals that depend on humid microhabitats. Their respiratory system, like that of all harvestmen, relies on tracheae, branching tubes that open to the outside through small pores on the body wall called spiracles, located between the bases of the 4th pair of legs and the abdomen. Unlike the book lungs of spiders and scorpions, tracheae provide no mechanism for preventing water loss, which confines these animals to perpetually moist environments. The most detailed account for the Sorensenellidae natural history to date was actually published in 1954, and there will be a link in the description to this document. It describes their food as consisting mainly of insects and other small arthropods. Under laboratory conditions, the individuals studied fed only on dead insects. However, the raptorial pedipalps of Sorensenella are almost certainly capable of seizing live prey as well. The architecture of the pedipalp, with its rows of inward-pointing spines forming a cage-like structure when closed, is a classic adaptation for grasping and restraining struggling prey. This is a predator's tool, not a scavenger's. It was also reported that all New Zealand Sorensenellinae lay small groups of eggs, typically 10 to 20 at a time, on the undersurface of logs and rocks. The eggs are guarded, and at intervals of a few days to a week, additional eggs are deposited, so that a single nest can accumulate 60 to 100 eggs in various stages of development. The arachnologist Forster described this as maternal care, a behavior well known in many arachnid groups. But he was wrong. In 2007, the Brazilian arachnologist Glauco Machado, working at the University of Sao Paulo at the same sex lab from previous videos, published a short but stunning correction. Machado noticed that in old photos, The guarding individual was unmistakably male. The photograph showed a male sitting over a clutch of eggs at multiple developmental stages, precisely the pattern described by Forster, but with the sex of the caretaker reversed. This was not a minor correction. Paternal care is one of the rarest forms of parental investment in the animal kingdom, and is almost unheard of among arachnids outside of the Harvestmen. Machado's interpretation represented an independent evolutionary origin of paternal care. In paternal harvestmen, the male defends a nest site and the females visit to deposit eggs before leaving. The male then guards the accumulating clutch, which may contain eggs from multiple females at different stages of development. He cleans the eggs, removing fungal hyphae and debris, and protects them from predators and from cannibalism by other harvestmen. The predators of Sorensenella are poorly documented, but the general suite of enemies facing New Zealand's ground-dwelling arachnids includes native and introduced species. Native predators likely include ground-hunting spiders, predatory beetles, and possibly the small secretive reptiles and amphibians of the New Zealand fauna. Introduced mammals like rats, hedgehogs, and mice are devastating generalist predators of forest floor invertebrates throughout New Zealand, and their impact on Sorensenella populations, while unstudied, is likely significant. Studies on other New Zealand harvestmen have confirmed that the European hedgehog and various introduced birds regularly consume harvestmen. The closest living relative of Sorensenella is Caramia, another New Zealand endemic genus. Genetic analyses have confirmed that Sorensenella and Caramia are sister genera, and have estimated that they diverged roughly 100 million years ago. Their geographic ranges appear to be complementary, with no known overlap. Where Sorensenella occurs, Caramia does not, and vice versa. In a broader taxonomic context, Sorensenella has relatives in Australia, New Caledonia, and South America. This distribution pattern is a sign of Gondwana origin. New Zealand began rifting from Australia and Antarctica around 80 million years ago, and the ancestors of Sorensenella were likely already present on the fragment of the continent crust that would become the New Zealand landmass. They did not disperse across oceans to reach their current home, rather the ocean formed around them. This makes Sorensenella a living relic. The New Zealand forests where Sorensenella lives are themselves ancient ecosystems, but they have changed dramatically over geological time. During the Oligocene, roughly 25 to 30 million years ago, much of New Zealand was submerged beneath the sea. This phenomenon was called the Oligocene drowning, and it may have reduced the landmass to a scattering of low islands. Whether Sorensenella's ancestors survived this bottleneck on remnant land or recolonized afterward is one of the great unresolved questions of New Zealand biogeography. More recently, the Pleistocene glaciations reshaped New Zealand's forests repeatedly, pushing tree lines down slope and fragmenting habitats into isolated refuges. For an animal as poorly dispersing as Sorensenella, each glacial cycle would have been a catastrophe, isolating populations that might never reconnect. The patchy modern distribution of the genus may be a relic of these ancient fragmentations. The limited dispersal ability of trianonichids is not just theoretical, it is visible in their genetics. Studies on related genera, particularly Algidia in New Zealand and Sclerobunis in North America, have shown that populations separated by even modest geographical barriers can accumulate millions of years of genetic divergence while remaining morphologically nearly identical. This pattern of deep cryptic diversity is likely present in Sorensenella as well, but no molecular population level study of this genus has been conducted. The conservation status and population size of Sorensenella is unknown. This is typical for harvestmen in general. They are too small, too obscure, and too taxonomically challenging for conservation agencies to care, yet their extreme habitat specificity, poor dispersal ability, and sensitivity to habitat modification make them exactly the kind of organisms that could be quietly declining without anyone noticing. The ongoing destruction and fragmentation of New Zealand's native forests, the spread of introduced predators, and the drying of forest microclimates due to edge effects are all threats that could affect Sorensenella populations.